all so much for coming today. My name is Natalie Love, and I'm currently a postdoc at Cal Poly in San Luis Obispo. But today I'm going to talk with you a little bit about some of the work that I did for my dissertation with my advisor, Susan Mazer. And what we did was we investigated region-specific phenological responses to climate and to climate change using herbarium specimens of Streptanthus tortuosus. So one of the major questions in ecology is how will climate change affect plant phenology? Because changes in plant phenology, whether that be the timing of leaf out or the timing of reproduction or even the timing of senescence, changes in all of those potential phenophases might have major ecosystem consequences. And so, for example, changes in flowering time has the potential to affect plant-pollinator relationships, especially if plants and their pollinators don't respond to climate change in the same direction. And it could potentially affect plant-plant interactions, such as competition for soil or for pollinator resources, or it could also potentially expose plants to novel environments, such as drought, or frost, or even novel herbivores. And so predicting how plants respond to future climate change or assessing how they've already responded to contemporary climate change is really a critical first step towards understanding the potential ecosystem-wide consequences of climate change. So one way that we can predict how species will respond to future climate change is to start by evaluating how species respond to current variation in climate using these types of models called PHENO-CLIMATIC models. And we can construct these models by looking at how the onset of some phenological event, such as flowering date, responds to variation in climate, such as temperature or precipitation, either from year to year or from location to location. So each one of the points on the graph here represents a phenological observation, and those types of phenological observations can come from field-based observational data, such as large-scale networks like the National Phenology Network. These observations could come from common and garden experiments, or they could be obtained from herbarium specimens. And so the slope of this relationship allows us to estimate the phenological sensitivity to a given climate variable, in this case to spring temperature. And it tells us that for every 1 degree Celsius increase in spring temperature, flowering date advances by about 4.2 days. And so if these geographic patterns in phenological responses are at least in part genetically based and potentially generated by natural selection, then evaluating these kinds of trait by environmental associations may help us make more informed or better predictions about the effects of climate change. And so this figure right here demonstrates a decline in flower color where these lighter flower colors are present in places that are warmer, so lower latitudes and lower elevations. And so based on this pattern, we might expect that as the climate warms, flower color might evolve to become lighter, especially if flower color represents an adaptation to warm temperatures. And that same kind of logic can be applied for phenology. So if earlier flowering in warm locations represents an adaptation to warmer climates, then we might expect flowering time to become earlier as the climate warms in the future. And so we may be able to use this kind of space for time substitution to try and predict or make more informed predictions about how phenology might change in the future. The only problem with this assumption is that the observed climatic conditions experienced by populations during a given year can really be partitioned into two components, and traits could potentially respond differently to these two components of climate. And I'm going to use temperature as an example to kind of demonstrate this concept and demonstrate what I'm talking about. So first, for any given population, there is a long-term mean temperature component. So throughout time at a given location, let's say this population, the temperature, here we have mean annual temperature, the temperature will fluctuate from year to year, but it kind of fluctuates around a mean. And variation among populations in their long-term mean temperature really represents spatial variation in chronic temperature, right? It's always relatively warm in the desert. It's going to be relatively cool in the mountains. And then second, there's this interannual component, which represents the deviation in temperature from a long-term mean during a single year or during the year that some phenological event was observed. And this is considered the climate anomaly. And climate anomalies can be positive, representing warmer than average years, and they can also be negative, representing cooler than average years. And so these two components of climate, the long-term mean as well as the climate anomaly, represent two pretty much independent sources of variation in climate, and meaning that oftentimes in California, they're actually not correlated with each other. And it's possible that traits like phenology might actually respond differently to these two different sources of climate variation. And these patterns might potentially be generated by different biological mechanisms. And so let's go ahead and take a look at a hypothetical example here demonstrating how flowering time among populations might respond to these two sources of variation in climate. And so here we see there's a negative relationship between flowering date on the y-axis and long-term mean annual temperature on the x-axis. And we see that populations in warmer locations tend to flower relatively early, and populations in cooler locations tend to flower relatively late. And the slope allows us to estimate the sensitivity of flowering date to this spatial variation in long-term mean temperature. And we might expect that this pattern or this decline in flowering date might be generated by a more long-term process. There we go. And so we might expect that this pattern or this decline in flowering date might be generated by a more long-term process, like perhaps local adaptation to chronic climatic conditions at the location where those populations are present. And here, among those same populations, we also see a negative relationship between flowering date, but this time we have the mean annual temperature anomaly during the year of flowering on the y-axis. And here we see that populations consistently flower earlier in warmer than average years. And again, the slope allows us to estimate the sensitivity of flowering date, this time to interannual variation in temperature. And so we might expect this pattern to be generated by short-term responses, so perhaps phenotypic plasticity or even very rapid evolution if deviations in climate are consistent from year to year, such as like a prolonged drought or potentially a period of warmer than average years like we've been experiencing during the past couple decades because of climate change. And so in some, species might respond to potentially one or both of these sources of variation in climate, and responses might differ either in the direction or potentially in the magnitude of their response. And so in this example, what I've done is I've demonstrated that this species here is more sensitive to interannual variation, meaning that the slope is steeper, more sensitive to interannual variation than spatial variation in temperature. So a 1 degree warmer than average year will advance flowering date more quickly than moving to a 1 degree warmer location. And so very few studies have investigated whether species respond differently to these two different sources of climate variation, but it could potentially be really important for predicting whether and how species respond to climate change. And we really also don't know which one of these estimates might give us a good sense of how plants will actually respond to future climate change. And if these estimates differ in terms of their quantitative sensitivity, those could potentially give us very different predictions about how quickly plants might respond to climate change. So part of my dissertation work was to assess whether flowering time responds differently, either in direction or in magnitude, to these sources of climate variation. And so many studies have demonstrated that plants especially are phenologically sensitive to temperature, but recent studies have shown that that sensitivity to temperature varies among species. And in this recent study by Park et al., each point here represents the estimated sensitivity of fruiting date specifically to temperature for some given species. And where points fall below 0, that means that species in warmer locations, species flower or fruit earlier in warmer places. And so in this study, they actually found that there is a geographic pattern to these differences in sensitivity, so that those species that are most sensitive to temperature, those species that have the greatest, where temperature has the greatest advancing effect on fruiting date, those species tend to be species that occur in warmer locations in these more southern latitudes. And so this leads to the question, well, what if a species was distributed across the entire eastern United States? Would then populations of that species be more sensitive to temperature in more southern latitudes? And so, in other words, does phenological sensitivity to temperature or to climate vary intra-specifically? And so there's been very few studies that have investigated these types of intra-specific patterns in sensitivity. So we really know very little about whether or to what degree it might occur within species. And this limits our ability to make kind of accurate predictions about how individual species will respond to climate change. And so part of the talk that I will give today will focus on investigating these types of intra-specific patterns of sensitivity in Streptanthus tortuosus. And so Streptanthus tortuosus, the California jewel flower or the mountain jewel flower, it provides a particularly good system for studying these types of questions and for evaluating the potential drivers of geographic variation in flowering time. First, it's very widely distributed in California. It occurs across a range of climates from more coastal areas to fairly hot, dry foothills, all the way up into the alpine areas of the Sierra Nevada. And so we might expect to be able to detect associations between climate and flowering date, or between the day of year of specimen collection and climate. And second, it's very well represented by herbarium specimens. There's over 100 years of herbarium records. There's over 1,000 records that are available in California. And this provides us with a really great data set to study these types of changes. So herbarium specimens provide us with this great snapshot of both the morphological and the phenological status of a plant at a given date at a very specific locations. And because they're so broadly distributed geographically as well as temporally, they allow us to evaluate responses of phenology to both long-term and short-term interannual variation in climate. And so using Streptanthus tortuosus as a study system, we addressed the following questions. So first, does reproductive phenology co-vary with long-term mean and or interannual variation in climate? And do these relationships differ in direction or differ in magnitude? And so answering these questions will help us understand whether phenology responds to potentially one or both of these different sources of climate variation, and whether or not those responses differ in direction or magnitude. And then third, I was really interested in understanding whether phenological sensitivity to climate varies within species. And because herbarium records are so spatially and broadly distributed, spatially and temporally broadly distributed, it provides us with a really great system to ask whether phenological sensitivity to climate varies within species, and whether this has potentially already led to divergent responses to climate change. So today I'm going to tell you two stories from my dissertation work using herbarium specimens to study reproductive patterns, reproductive phenology in Streptanthus tortuosus. So first, I am going to tell you a little bit about how Susan Mazer and I worked to develop a new metric to improve the fit and the predictive power of phenoclimatic models that are designed to estimate the sensitivity of flowering date to various climate variables. And I'll talk briefly about some of our recent collaborative work with some colleagues in France using deep learning to score herbarium specimens in order to estimate their phenological status. And then second, I will talk about how we used phenoclimatic models to investigate whether phenological sensitivity to climate varies within species, and whether there have already been shifts in flowering date that have been captured by this 100-year or so herbarium record. So we can go ahead and start with the first story. And I wanted to start by talking a little bit more about how we actually use herbarium specimens to construct these phenoclimatic models. And like I said before, herbarium records provide us with this really great snapshot of plants back in time at a given location, and all of the information that we need is provided on this label here. And we can use these herbarium specimens to construct these types of models. And so, for example, this is an herbarium specimen that's housed in the California Academy of Sciences. It was collected by my personal botanical hero, Alice Eastwood. And we use this specimen in our larger data set. And so first, we know that this individual was flowering when it was collected, because we can see that there's actually flowers on the specimen sheet when it was collected on July 25th in 1923. And we can turn that collection date into a day of year that ranges from 1 to 365. And this day of year of collection is oftentimes used as a proxy for first flowering date or for peak flowering date in these types of studies. So I have just flowering date here written on the y-axis. And then second, we know where this specimen was collected, and we can use that information to extract climate data for that location in the year that the specimen was collected. And there are multiple places that we can get that climate data, either from PRISM or Climate NA or CHELSA, and we have that here plotted on the y-axis. And so these two pieces of information can be collected for many herbarium specimens, and especially for those with really good collection information. And this allows us to quantify the relationship between flowering date and any climate variable that we think might be important for cueing the onset of flowering. But herbarium specimens, they're oftentimes collected at any stage really between all buds and actually fruiting, especially in Streptanthus tortuosus, which has fairly showy fruits and therefore oftentimes is actually collected in fruit. And so this variation among specimens in their phenological status or their phenological stage can potentially introduce noise or bias into these types of phenoclimatic models. And just to give you an example, this specimen here, that specimen might have been collected later than we might expect based on the temperature where it was collected, because maybe it was collected at a really late phenological stage. Maybe it was actually collected in fruit. And this type of noise or this type of variation among specimens, it could also potentially introduce bias into these types of models. And just to give you an example of that, these specimens here, maybe those specimens that are collected in cooler locations, maybe they're actually just collected at later phenological stages because they're more difficult to get to, right? These are herbarium specimens that are collected by human collectors. And so maybe the roads were closed because it was very snowy and these were difficult to get to. So they were actually collected at later phenological stages. And so this type of noise or bias, it could potentially make it harder to detect a relationship between flowering date and climate, or it could potentially limit our ability to kind of accurately estimate their phenological sensitivity or estimate the slope of this relationship. And so some studies have dealt with this variation among specimens in their phenological status by using specimens that meet a certain criteria, like specimens that are in flower or specimens that have greater than 50% flowers. But that can potentially limit the sample size, and that could be problematic, especially for species that are not as well represented by herbarium specimens. And so Susan Mazer and I worked to develop the phenological index, which is a quantitative way to control for variation among specimens in their phenological stage when constructing phenoclimatic models. And so the phenological index is an integrative metric that is derived from the proportion of reproductive structures in a given class multiplied by the value of that class that represents the degree of phenological progression. And so for Streptanthus, we ended up using 4 classes. Buds were given a value of 1 because they're the earliest phenological phase. Flowers were given a value of 2. Immature fruits were given a value of 3, and mature fruits, the last phase, were given a value of 4. And so a specimen that had all buds would have a phenological index of 1, and a specimen with all mature fruits would have a phenological index of 4, and we could have any potential number in between depending on the proportion of reproductive structures in each one of those classes. And this index can potentially be useful for other species as well. It's adaptable and flexible in terms of the number of stages or the number of classes that can be included. And so some members of the Maser lab were using this metric to score specimens of Nemophilum and Zizii, and they actually ended up using 5 classes in it, and it worked out really well for them. And so this is an example of one of the specimens that we scored. The specimen has 97 buds, 55 flowers, 16 immature fruits, and 29 mature fruits. And when we use that to calculate its phenological index, we end up with a PI value of about 1.88, which means that it's generally kind of right in the middle of its phenological progression. And so as you can imagine, it's difficult to keep track of all of the counts of potentially hundreds of reproductive structures present on each one of these herbarium records. And so we developed a kind of fun way to keep track of these counts using ImageJ. We were using all imaged herbarium specimens. So we used an ImageJ plugin called Cell Counter, which was originally designed to help scientists keep track of where cells were in images. And we were able to adapt that and use it to keep track of the number of reproductive units on each one of these herbarium specimens. And we can place a little colored marker of a given class on each one of the structures, and then ImageJ will keep track of those counts for us. And the nice thing about this is it actually saves the XY coordinates of the markers, and that can be recalled later. And I will talk in a little bit about how that was really useful data for some of our collaborators to have. And so for this study, we wanted to use a small subset of the herbarium specimens of Streptanthus tortuosus to as a test case to demonstrate how the phenological index can be used in phenoclimatic models. And specifically, we wanted to know, does including the PI in phenoclimatic models as a main effect improve their predictive power? And second, we wanted to know, does including the PI change the estimated relationship between flowering date and climate? And so we used our ImageJ protocol to score a small subset, about 119 specimens of the 1,400 or so imaged herbarium specimens that were available to us. And we georeferenced all of those specimens and then used that location information to download site and year specific climate data for each specimen from PRISM. And specifically, we downloaded the spring maximum temperature and the winter cumulative winter precipitation, which are 2 climate variables that have been demonstrated to be related to phenology in other species. And then what we did was we constructed 2 multivariate linear regressions to investigate or to quantify the relationship between flowering date and climate. And one of those models included the phenological index as a main effect, and one of them did not include the phenological index. So let's take a look at some of the results here. So here we are looking at the proportion of variance explained by each main effect in first the model that did not include the phenological index as a main effect, and then the model that did include the PI as a main effect. And what we can see is that including the phenological index as a main effect reduced the amount of unexplained variance by explaining about 11% of the variance in the day of year of collection. And the cross-validated model R-squared increased from 21% to about 42%. And so overall, including the phenological index in these phenoclimatic models reduced the unexplained variance, reduced the model error, and increased the model R-squared, or the predictive power of the model. Okay, so next let's take a look at the quantitative relationship between the day of year of collection and climate for both models. So here we're looking at the partial regression plots showing the relationship between DOY and spring temperature and winter precipitation. And what we can see is that flowering time advances about 4.2 days for every 1 degree increase in temperature. So plants, individuals flower earlier at relatively warm sites and later at relatively cool sites. And then we actually see that increased precipitation delays flowering date. So individuals are flowering relatively early in areas that are warm and dry. And when we look at the partial regression plots for the model that does include the phenological index, what we can see is that the variation around the regression line in both of those relationships is reduced, especially in the temperature relationship, but the estimate of the slope actually remains the same. So the slopes are actually estimating the same sensitivity to those climate variables. So including the phenological index improves the model fit by accounting for variance in DOY, but it doesn't seem to actually change the estimated relationship between phenology and climate, at least in this subset in this species. But the parameterized model that actually does include the phenological index as a main effect can then potentially be used to actually predict the day of year of onset of any phenophase at any given set of climatic conditions. And so we could potentially do that by plugging in a specific value for PI here. So for example, a PI value of 2 might represent peak flowering. And so we could potentially predict the day of year of onset, specifically of peak flowering or of fruiting under a specific set of climatic conditions. And we also have a slope estimate for the phenological index. And as we might expect, it behaves as we might expect, which is that specimens that are collected at later phenological stages, such as fruit, those specimens are collected at later DOIs as well. And the slope of this relationship actually allows us to quantify the length or estimate the length of the reproductive period. And so this estimate tells us that it takes about 17 days for a plant to go from a PI of 1 to a PI of 2. So for a plant to go from budding to flowering. So for a plant to go from budding to fruiting would take an estimated length of about 50 days or so. And this could be potentially really useful because it would allow us to test hypotheses regarding how spatial variation in climate could potentially drive geographic variation in the length of the reproductive period. And so, for example, we might predict that collectively, maybe individuals collected in alpine regions might progress more quickly through reproduction because their growing season is shorter. And so it might seem that counting potentially hundreds of reproductive structures on a sheet would be a really tedious task to do for a single species, let alone extend that to thousands of species represented by millions of herbarium sheets that will soon be available in California alone. But it's possible that we might be able to use some machine learning or some deep learning to do some of this work for us. And one of the great outcomes of this study was we were able to take our scored set of specimens and then work with some collaborators who could then use them and use the XY coordinates of the scores that we added on ImageJ. They use them as a training data set to develop machine learning algorithms to automate the detection of the different types of reproductive structures for Streptanthus. And so in this study that we recently published, we scored about 100 specimens using a few different types of machine learning algorithms, but the most successful, this full mask algorithm shown in red, was able to correctly detect and classify reproductive structures about 78% of the time, and was trained using only 21 scored specimens. So it has the potential to speed up or automate the detection or the counting of reproductive structures on herbarium specimens. And since this paper was published last year, we've been working more, and we've been using this full mask algorithm that we trained and developed to actually score a larger data set of about 700 herbarium records that we had also scored by hand with the help of some undergraduates. And we could then we then wanted to be able to compare how well the machine learning algorithm scored specimens compared to what we counted by hand. And so here we're looking at some of the results from those comparisons. So we have counting error here on the y-axis, and counting error indicates how well the algorithm can actually classify and count reproductive structures. Specifically here, we're looking at flowers. So 0 counting errors means that the algorithm did a good job. It counted the same number of flowers per sheet that we counted by hand. And these negative counting errors mean that the algorithm counted less than what we counted by hand, or it underestimated the number of flowers present on each sheet. And we were really curious to know whether the counting error actually depended on the number of structures present on each sheet. And so does the algorithm do a better job of detecting structures when there are actually fewer of them present on the sheet? And what we can see is that when there's kind of 0 to 19 flowers on a sheet, the algorithm does a pretty good job of counting them, but as there's more structures on the sheet, the algorithm tends to underestimate the number of structures present. But it seems to underestimate the count of each class of reproductive organ, flowers and buds, in very similar proportions. So when we actually use those counts to calculate the phenological index, which relies on those proportions instead of the raw values, when we use it to calculate the phenological index, what we calculate by hand is very similar to what is calculated using the machine learning algorithm. So they're highly correlated with each other. So using machine learning, we were able to accurately derive the quantitative estimate of the phenological status for each herbarium record. All right, so next I'm going to spend a little bit of time talking about when we used a much larger data set of herbarium records, and we were able to assess intra-specific variation in phenological sensitivity and responses to climate change. And so, like I mentioned, one of the major goals of ecologists is to be able to predict how plants will actually respond phenologically to climate change, because this could help us better understand how species interactions might potentially be impacted by climate change. And one way that we can make these predictions is to estimate species' phenological sensitivity to climate. And like I said, we know that sensitivity to climate varies among species, and that, for example, in this study, there could potentially be a geographic pattern where species that occur in warmer, more southerly latitudes are more sensitive to temperature than those in cooler, more northern latitudes. And so the authors of this paper actually predicted that, all else being equal, these species that occur in warmer locations would advance their fruiting phenology more quickly, meaning they'll start to fruit earlier in response to climate warming because they're more sensitive to temperature. And like I said, we really don't know much about whether or to what degree phenological sensitivities vary intra-specifically or within species, and whether this might have already caused divergent phenological shifts within species, which could potentially not only disrupt the species interactions like I talked about, but also has the potential to disrupt gene flow among conspecific populations. And so, taking inspiration from this paper, we first wanted to test whether flowering time has already shifted in response to climate change across the range of Streptanthus tortuosus. And we wanted to test whether these shifts differ specifically in warm versus cool regions of the species range. And so if we were able to detect kind of divergent shifts in flowering time, we wanted to test two potentially non-mutually exclusive drivers of these shifts. So first, divergent shifts could potentially be caused if individuals collected in the different regions have differences in their phenological sensitivity, either to long-term mean or to interannual variation in climate. It's also possible that climate warming has not occurred at the same rate across the entire range of the species. So it's possible that some regions might be warming faster than others, and this could also potentially drive divergent shifts or explain divergent shifts in phenology. And it's possible that both of these non-mutually exclusive drivers are happening at the same time. So there could be regional variation in sensitivity, and there could also be regional variation in climate warming. And these could potentially kind of work synergistically in driving divergent shifts in flowering time. So to address these questions, we used a data set of about 750 imaged and scored herbarium specimens. And these specimens were collected between 1902 and 2013. So we had a 112-year collection record of Streptanthus tortuosus. And they were collected across a fairly wide range of elevations and therefore a fairly wide range of climates in California. And so we extracted the day of year of collection, and we georeferenced each one of these specimens, and we used those locations to download the site and the year-specific climate data from Climate NA. And we downloaded a range of seasonal and annual variables, but today I'm mostly going to be focusing on winter and spring and summer temperatures, which showed kind of would demonstrate the general responses that we detected in Streptanthus. So first, what we had to do was categorize our specimens as occurring either in chronically cool or in chronically warm regions. And so what we did was we first divided them into two groups based on their median mean annual temperature. And then to make a stronger comparison between regions, between warm and cool regions, what we did was we actually removed the middle 20% of the specimens that are in warmish, coolish areas. And those specimens are shown here in white. And because our data set spans over a century of collections, we can ask whether the day of year of collection has changed through time and whether the shift differs between regions. And so we estimated these effects by looking at the relationship between the day of year of collection and the specimen collection year. And we included this year by temperature region interaction to ask whether the relationship or the potential shift between year and day of year of collection differed in warm and cool regions. And like I mentioned, we scored all of these 750 herbarium specimens for phenology, and we assigned all of the specimens a phenological index. And so all of our models include the PI as a main effect to control for variation among specimens in their phenological status when estimating these shifts through time. And we also used linear regression models to estimate the sensitivity of the day of year of collection to both long-term mean and climate anomalies for each one of these climate variables. And we included a climate by temperature region interaction, again, to ask whether sensitivity to climate differed between regions, whether sensitivity to climate varied intra-specifically. Okay, let's take a look at some of the results that we found. So here we're looking at the relationship between the day of year of collection and year. So looking at how the day of year of collection has changed through time. And first, we did indeed find that there have been divergent shifts in flowering time during the past 100 years. So individuals in warm regions have advanced their flowering date by an estimated about 19 days. So they're being collected about 19 days earlier than they were about 100 years ago. But we found no significant advancement of individuals collected in cool regions. And so this divergence or this difference in the rate of advancement through time could be because potentially maybe individuals in warm regions are more sensitive to temperature, or it could be because that they've experienced a greater increase in temperature during the past century, or it could potentially be both of these factors together. So first, let's take a look at how a sensitivity to climate differs between regions. And again, I'm going to focus specifically on winter temperature and spring and summer temperature, which are the seasons during which Streptanthus is developing buds and actively flowering. And we might expect that temperatures during these seasons might be important for Streptanthus in terms of cueing flowering. And so here we're looking at the relationship between the day of year of collection and the long-term mean or the 90-year mean winter minimum temperature. And in both regions, individuals flower relatively early in warm locations, but individuals in warm regions are more sensitive to the spatial variation in temperature than individuals in cool regions. And when we take a look at the winter temperature anomalies during the year of specimen collection, we see that individuals in warm regions are sensitive to the anomalies, but individual in cool regions are actually not sensitive to anomalies. They're not adjusting their flowering date or changing their flowering date in response to warmer or cooler than average years. But in warm regions, individuals are consistently flowering a bit earlier during warmer than average years and a bit later during cooler than average years. And so this is interesting because it might mean that these interannual deviations or anomalies in winter temperatures, maybe they might occur just too early for individuals in cool regions to really respond to. They are flowering generally a little bit later. They're flowering in June and August, so maybe the temperatures in winter don't have as much of an effect on their flowering date. But on the other hand, Streptanthus does have a vernalization requirement. So it needs a certain amount of chill in order to induce flowering. And if the chilling hours are actually accumulating during winter, then we might expect individuals in cool regions to be sensitive to these winter temperature anomalies. And in fact, we might even expect that warmer than average winters might delay the accumulation of chilling hours and might also delay flowering time. So another interesting pattern to note is that in the case of both regions, the sensitivity to long-term mean temperature differs compared to the sensitivity to interannual variation in temperature. So the magnitude of those relationships between long-term mean and climate anomalies does differ. Okay, so I also want to take a look at the sensitivities to spring and summer temperature when plants are actively flowering. And here we're looking at the relationship between day of year of flowering and the 90-year mean spring and summer minimum temperatures. And these patterns are similar to the relationship with long-term mean winter temperatures. So in both regions, individuals flower relatively early in warm locations, but again, individuals in warm regions are more sensitive to the spatial variation in temperature than individuals in cool regions. And in the case of spring and summer temperature anomalies, in this case, individuals in cool regions are sensitive to the spring and summer temperature anomalies, so that a warmer than average year will actually advance flowering date in cool regions, and it will also advance flowering date in warm regions. However, individuals in warm regions, again, more sensitive to this interannual variation in temperature. So we've demonstrated that Streptanthus tortuosus is sensitive to spatial variation in temperature, and it's also sensitive to this interannual variation in temperature or these temperature anomalies. But more importantly, we found that individuals in warm regions are about 2 times as sensitive to both of these measures of sensitivity, so both long-term mean and interannual variation in temperature. And this difference in sensitivity could potentially alone explain this divergent phenological shift that we detected, but I also wanted to take a look at how temperature has been changing through time in these different regions and whether it has changed consistently. And so here we're looking at the relationship between minimum winter temperatures across the 100-year collection period. And what we see is that there's been about a 1 degree increase in winter temperatures in cool regions, but about a 2 degree increase in winter temperatures in warm regions. And we see a somewhat similar pattern when we look at spring and summer temperatures. Temperatures increased in both regions, but it's increased more quickly in warm regions than in cool regions. So there's been a greater increase in temperature during the past 100 years, and these individuals have experienced potentially up to a 3 degree increase in temperature in the spring and in the summer. So in conclusion, we found that there are regional differences in phenological advancement through time. And we found that these regional differences in phenological advancement through time, they could be potentially driven by both sensitivity to temperature and the magnitude of climate warming. So individuals in warm regions are more sensitive to temperature, and individuals in warm regions have also experienced a greater degree of warming than individuals in cool regions. And so this study really demonstrates that we need to consider that potentially both regional differences in sensitivity and regional differences in the magnitude of climate warming could contribute to regional shifts in phenology that have already happened or could potentially happen in the future as a result of continued climate change. And it also shows that phenological responses to climate and to climate change might be more spatially complex than we really had previously thought about, and that it can also potentially vary intra-specifically. And so to accurately forecast phenological responses to future climate, we might need to consider using more fine-grained estimates of phenological sensitivity. So maybe region-specific sensitivities to account for some of the spatial complexity in responses. And so consequences of these shifts in flowering time are hard to predict. Phenological shifts have the potential to disrupt species interactions, but without the full picture of how other species are potentially responding to climate change, it's difficult to kind of make concrete predictions about the realized consequences of these shifts for species interactions. Okay, I have just a few thank yous to give out. So I wanted to thank my lab mates who I've had great conversations about this research with, undergraduates who did a lot of the meticulous herbarium specimen scoring for us, the funding researchers or the funding resources that helped support my work throughout my PhD, and our collaborators in France that have been working with us on the deep learning and the machine learning. And then, of course, the top 10 collectors of Streptanthus tortuosus. Without them, I would not have been able to do any of this research. And I saw that one of them is actually here tonight. So I'm glad that I got to share that research with him. So thank you guys all for listening. And I think we got a few minutes for questions. I'm happy to stick around and answer any questions that anybody has.
That was amazing. Thank you, Natalie.
Yes, of course.
I'm pleased to say that we got the talk recorded because I'm gonna need to go back and listen to it again.
Okay, great.
It's not that I didn't get the take-home message, but I really want to sort of sit down and get into it. So, does anyone have any questions? You can unmute yourself and ask questions.
Or I can also keep an eye on the chat too, see if anybody has any questions.
Natalie?
Yes.
I have just one question. This may be off the wall, but has anybody ever tried measuring this phenological index or something like it in a real population? And maybe as a standard, maybe as a test, but rather than using herbarium specimens to actually, if I'm just envisioning a relatively reasonably sized population where you mark a few plants and you actually follow it, phenological changes plant by plant. Has anybody done that? And I don't know if it would be that valuable, but I can envision doing it in a few regions, older regions.
I can't think of any that I've read. It would be useful in the sense that I talked about how we could potentially estimate the rate of phenological progression. And so it would be really useful to go out into a population and do that and, like take a snapshot of the phenological index through time and see actually how quickly plants are progressing through their phenology to try and see if our estimates from herbarium specimens are at all similar. But when Susan and I were talking through the index, we were thinking that it could also be useful just for observational studies from phenology networks where people are kind of sporadically going out and collecting phenology data. If there was some kind of way to use the phenological index, you could use it on live plants. You could take the same counts and use it in a similar way even with live plants.
I mean, it would be extremely tedious and difficult, and you would need to get visit the population once a week for a while or something like that. Somebody on the chat said they've done it for Dicentra uniflora. Nice. So anyway, just curious about that. It might be fun to try.
Yeah, and Natalie, I have data from Pepperwood Preserve for the entire community for 4 years with buds, flowers, and fruit counted for all species. So we could do that with that data set.
That would be really interesting. How was that data to collect?
It was tedious. Yeah, I just went out to plots every week, and some days in the peak of, flowering, I actually counted all grass flowers also.
Yeah.
So it was pretty intense during some of those days, and some of the counts went into 2-day counts. Yeah.
Oh, jeez.
Wow.
Hi, Natalie. Nice talk. I enjoyed it, and some of your conclusions were right on the mark, especially the business about not being sure whether there are different, say, ecotypes that have different responses.
Yeah.
One of the things I've found from looking at many populations of Streptanthus tortuosus is that we may not be looking at one species.
Yeah.
I mean, we may be looking at a number of, at the very least, ecotypes or subspecies. And I'm just wondering how much that might influence your results. So for example, I've looked at some populations in the Klamath River, the Trinity River watershed, where one ecotype blooms perhaps a month earlier than the other, which would would be sort of interesting.
Yeah, that is really interesting, and I have been wondering about that, because going through all of their herbarium specimens, I see like they used to be subspecies something else, and I know that there's a lot of variation in terms of ecotypes within the species, and so I wonder if some of the differences in sensitivity that I detected are actually due to kind of like very, very different ecotypes of Streptanthus. And so it's if we did it it would be really interesting to do at a finer scale. I did this very kind of coarse, like warm versus cool comparison, but it would be interesting to do at a finer scale to see if you could potentially pick up any ecotype differences. And in my data set, I do have, I didn't actually do any analyses on it, but I do have a column with the subspecies names on there. And it would be interesting to kind of split the data set up into the different subspecies to see if there's any differences in sensitivity or in shifts.